Hey everyone, I'm Ruben. I'm going to be your moderator today for this episode of Redtalks. Just to introduce myself, I spent most of my career doing machine learning and big tech. Currently, I play a few different roles more in the tech bio ecosystem as an advisor, angel, and founder. Um, Before we get started, I just wanted to thank everyone for tuning in today, as well as the Kaleem Institute for organizing everything. And as a reminder, everyone tuning in can use the chat and Q&A box to share questions and comments throughout the session. In this episode, we're going to hear about some advancements in kind of different alternative approaches to computational methods in a couple different application areas, including kind of mechanistic methods for digital twins in trial design to AI-based prognostication and treatment response prediction. To take us through their work on these topics, we're glad to welcome Dr. Iraj Hosseini and Dr. Isen Naiken. Uh, Dr. Iraj Hosseini is a distinguished scientist at Genentech, where he leads work in modeling, simulation, and translational PK/PD. He focuses on using digital twins and computational models to better understand disease and improve clinical trial design. Dr. Isen Naiken is a PharmD and PhD in informatics and artificial intelligence at Université de Bourgogne. He's also co-founder and CSO of NexoMedis and editor-in-chief of Revue IA Santé, with a focus on AI applications in healthcare and oncology. Together, they're going to explore how AI and digital twins are changing the way we discover biomarkers, understand treatment response, and design the next generation of clinical trials in lymphoma and a little bit in multiple myeloma. So just to go into some of the high-level kind of comparison of the methods we're going to be seeing. So as kind of a high-level introduction and reminder, we're going to be going over some mechanistic methods with Dr. Hosseini. And this is more classical and more interpretable as a kind of modeling approach where you're kind of directly encoding model parameter relationships based on kind of known biology, known medicine, and everything like that. The benefit to this is really you're kind of doing high bias modeling with low variance that allows you to be very data efficient. You can do interesting things with small amounts of data, and you can kind of like interpret and question the validity of the model based on the actual model parameters themselves, among other things. To contrast that, we have kind of the AI ML approach, which is much more data-driven. So the general approach here is that we don't make any real assumptions in the model parameters themselves. And this kind of puts emphasis more on the data itself. So in how does the model fit? What does the training data look like? And does the training data kind of capture the different relationships that we are trying to model? And so a lot of the like questioning and reasoning around those models are also kind of focused more towards around what are the statistics around the training data set? How is that model actually validated in the holdout scenarios, and how do those scenarios relate to, something I care about? And so with that, I'll hand it over to Dr. Hosseini to talk about mechanistic models for trial design.
Thank you, Ruben, for the kind introduction. So let me go ahead and share my screen. So it's a great pleasure to be here to talk about a little bit about the work that we've done for QSP in the loop and digital twins in the past 7-8 years. And then how we are leveraging digital twins to enhance decision-making in drug development. And I want to sort of talk about a couple of cases because that's the best way to essentially talk about these applications. So what's the digital twin? It's a buzzword that we hear in a lot of different industries, a lot of different applications, a lot of people are working on it. And in fact, I think what I thought would be useful is to look at an example from NVIDIA Omniverse. They have developed these platforms that can essentially create a digital replica of Amazon warehouse. And there are some learnings here. So to introduce this, I'm going to click on this video for a couple of minutes to see how they are defining digital twins.
Every day, hundreds of Amazon facilities handle tens of millions of packages, with more than two-thirds of these customer orders handled by robots. To support this highly complex operation, we deployed hundreds of thousands of mobile drive robots and associated storage pods, which allow us to store far more inventory in our buildings than traditional shelving, and which help us move inventory in a safer, more efficient way. Key to the scaling has been our ability to simulate these buildings and understand their performance before we build them. Let's take a look at how NVIDIA Omniverse is helping us optimize and simplify these processes. At Amazon Robotics, we're able to create full-scale digital twins of our warehouses in NVIDIA Omniverse, helping us optimize warehouse design, train more intelligent robot assistants, and gain operational efficiencies. In Omniverse, we are uniquely able to aggregate datasets from many different CAD applications and visualize these massive models in full fidelity realism, enabled by Omniverse's RTX-accelerated ray tracing, materials, and physics. Digital twins are an integral part of future warehouses and factories, enabling continuous integration and continuous delivery. With each new software and layout optimization, we can test in the digital twin before releasing to the physical warehouse, preventing any system downtime or failure while maximizing operational efficiencies.
Okay, so there are a couple of lessons that we can learn from here. First of all is the massive scale of like these Amazon warehouses. And the fact that they are probably working at a very high performance, but like if you want to introduce a new routing optimization or a new thing, it's hard to implement it physically because the cost is huge, right? So in this case, they can test it in the software, they can look at the pros and cons, and they can see in these situations, you're probably going to gain a few percentages, and in these situations, you're going to get some sort of like downturn, but essentially that at the end of the day, among the different routing optimizations that you may have a higher expected value of efficiency and therefore introduce it. So that's sort of like the way that this would work for Amazon warehouses. But to think of it is, first of all, they had a model. They had a model that reliably could work and essentially reproduce what's happening in Amazon warehouses. And then second thing was to test that and personalize it against each of the Amazon warehouses that they have. It doesn't matter whether it's in Seattle or Toronto or somewhere else, but basically personalize it to that Amazon warehouse. And these are the two things that we're going to essentially keep seeing in these presentations. So now moving on, when we are building biological digital twins, we have a lot of information. So like when we're sort of like building a digital twins for Amazon warehouses, we have a lot of information. We know exact position of the shelves, the way that robots play, the speed of those things, and sort of like the material properties that we see in these warehouses. But when it comes to building biological digital twins, you're pretty much looking at an Amazon warehouse from 30,000 miles. You don't know what's inside. Pretty much what you see is a little bit of an input and output, how many workers are going there, and sort of like how many boxes are coming out. That's why one digital twin would not work in a biological digital twin, given that there is a lot of uncertainty and there's a lot of unknown that we don't know about cells, about mechanisms of actions, about like how things interact with each other, and sort of like the heterogeneity that we have in patients. So now that's another point that differentiates a biological digital twin from a digital twin that you are hearing in other fields. So now I asked ChatGPT, what is a QSP digital twin, quantitative systems pharmacology? First, it generated this image. Obviously, it's a bit scary. I asked to try again, and we got this image. And I think this is a little bit more meaningful. So assuming that we have a model of a specific disease, a digital twin is a set of virtual subjects or different parameterization of a model that can accurately reflect an individual patient. And I think that's very important for us. Accurate enough is where we are going with this. Obviously, you're not going to have all the information, but you're going to have enough information to answer the question of interest. Okay, so let's start with a specific example because I think that's going to make it more real. This audience probably is familiar with T-cell dependent bispecific antibodies. These are antibodies that have two different arms binding to each other. One arm binds to CD3 on T cells, the other arm binds to the target cell on the cancer cells, and it brings the T cell and cancer cell together. So in this case, I'm going to talk about mosunetuzumab, which is a CD20, CD3 bispecific. CD20 is expressed on B cells. So in this case, the T cells get close to the B cells, they get activated, they kill the B cells, and at the same time, they release cytokine. The cytokine release syndrome is the on-target safety, and then the efficacy is killing of the tumor cells. This is a very promising therapeutic modality. It's been utilized in heme indications, particularly in follicular lymphoma, DLBCL, NHL, as well as some other targets in multiple myeloma. So 7, 8 years ago, when we were developing this drug, the question was how to optimize the dose so that we can minimize the cytokine release syndrome as well as maximizing the efficacy. So to answer that, we built a model. This is a fairly complex model of the interactions between the B cells and the T cells throughout the immune system. It has multiple compartments: peripheral blood, bone marrow, lymph node, spleen, and the tumor. It has the way that the T cells get activated in the presence of the mosunetuzumab or other TCPs. They get activated. The activated T cells proliferate. They kill the B cells. They get deactivated or partially de-exhausted, go back to resting cells. At the same time, they release cytokines, as well as they traffic between the different compartments. So given that the model is fairly complex, we used a very diverse set of data in order to constrain this model. First of all, our fundamental understanding of immunology, the in vitro potency data that we had generated for mosunetuzumab, the rich data set that we had in cyno monkeys in terms of PK and PD, sometimes even tissue biopsies, the human pathology, as well as at the time we had data from belantamab, which is yet another biospecific. It's targeting CD19 instead of CD20, but it has very similar mechanism of action. And later on with data from our drug in phase 1, in order to answer that question. So we basically came up with a step-up dose regimen that suggested that if you use that approach, we can reduce CRS, but also not impact the efficacy. Now, we tested that drug, and essentially that approach worked. And we got to the point that now we had quite a bit of data in our phase 1, about like 35 patients. And what I'm showing here, each color is a different cohort. They've received an increasing dose. When you go from left to right, we have increased the dose. And when our physicians were looking at this, they wanted to know what is the optimum dose to use. And obviously, when you look at this picture, it's very unclear. It's very messy. It's very noisy. And you could have a scenario that you would look at the patients in the red cohort here and say all 5 patients responded in this cohort. They all have about 50% or more reduction in tumor size. So therefore, this is the best dose to use. Whereas I think the argument that we had at the time was this data is not complete. There is an element of luck in this. And we cannot really just use the limited data in order to answer this question. In fact, let's use all the data that we have in order to see if we can come up with a better picture. So the challenge that we had was data was available, but they were available from multiple dosing regimens, flat dosing versus double step. The small size of cohort, maybe n equal 1 to 7 per cohort. The tumor measurement time point variability, right? So typically you measure that 2 cycles after the treatment, so 42 days, but sometimes it's like 56 days, sometimes it's 36 days. So you have that variability and quite a bit of patient heterogeneity. The baseline characteristics were different, the disease types were different, and all these patients, because they were relapsed refractory, they also had CD20 targeting treatments in their system, previously treated with rituximab or other CD20 targeting agents. So given all these, we wanted to incorporate them into our system. So we came up with a novel workflow to generate QSP virtual population, which represented the variability in biological, pharmacological, and tumor-related parameters for our trial. And we call them digital twins. So how does it work? Our system already has quite a bit of variability incorporated in it. The things that we've seen preclinically, as well as sort of like some of the data that we had from Blin. But now, for any given patient, so I'm starting in 1A, I pick one patient, and I generate about 10,000 different patients with the variability that I assume in the tumor microenvironment. What is the size of the tumor? What is the proliferative capacity of the B cells? What is the B to T cell ratio? How fast the drugs can get there, and then so on and so forth. So all the gray lines are all the potential digital twins for a specific dose. Then we pick the exact time points and samples that we have for a given patient. Typically, you have the baseline and 1 or 2 measurements after. And we only pick the digital twins that match those two. So now you essentially have 50 or 100 patients or digital twins that match your exact time points for a given patient. Okay, so let's assume that we are having 40 patients and we are picking 50 digital twins per patient. That basically creates now a 2000 digital twin virtual population that you can run it in silico and test the different scenarios that you want to test. So now you can imagine that once you put things together here, you can reconstruct the waterfall plot for each of the doses. For example, you can see even though in some of the scenarios we didn't have that many responders, the model actually suggests that it was just an element of luck. And if you had incorporated or if you had recruited more patients, you would have seen patients responding. Obviously, we tested this just to essentially make sure that we have validation. So we also simulated our patients and mapped it against the data that we have just to make sure that the median of our virtual population goes through the median of the data, 25 to 75 goes through that, and 5 to 95. So that's a soft validation that our digital twins are not an overfit to the data that we have collected. Okay, so what would be one of the first applications that we'll have here? I would guess that the audience is familiar with waterfall plots. So here we also did the same thing. We wanted to simulate our digital twins to predict the percentage of the patients that respond to mosunetuzumab. So here you can see the doses that I'm simulating here is a step-up dosing, obviously, but the target dose is 2.8, 13.5, 27, and 60. And the question at the time was, we were testing 13.5. So you can see from 2.8 to 13.5, you get quite a bit of efficacy and improvement in the number of patients that respond. But as you increase the dose to 27 and 60, what looks like is happening is you're not getting more responders. So essentially the dose response relationship is saturating. You are reaching a plateau and you're not getting enough bang for your buck as you're increasing the dose. In fact, it may increase your safety. So the recommendation from our model at the time was it's worth to try 27 and 60, but it doesn't make sense to go higher than that, given that we are saturating, and that might be a good place to take these doses and go as sort of like recommended phase 2 dose and test it in a bigger expansion cohort. Lo and behold, that dose has now been approved as the dose for mosunetuzumab for the communication we had with the FDA. So the dose of mosunetuzumab is 1, 2, 60, 60, and then after that it reduces to a 30 maintenance dose. So this is now essentially showing that models like this can guide you in your design and sort of like as you are thinking of your clinical trial of when to stop and then when you expand your expansion cohort. Other things that you can try is also answer how many patients do I need to enroll in my clinical trial in order to see. This is essentially just sampling the populations and see what would be the number of patients that would give you a probability, like for example, an 80% probability in order to see a specific ORR that you have for your clinical design. One other aspect that we use this is essentially to help research questions for version 2 programs. What are the differences that we are seeing between version 1 and version 2? And sort of like, what are the things that we want to design in the molecule if we are trying to improve the efficacy? So we looked at the responders versus non-responders in our digital twins. And we looked at a lot of different parameters to see if there are differences between them. Specifically tumor parameters, B-cell killing parameters, and then T-cell activation. As we compared the responders versus non-responders, we saw that in terms of the B-cell killing and T-cell activation, there is really not that much of a difference between these two populations. But when you look at the tumor parameters, there's two big things. One is these patients, the non-responders tend to have a higher proliferative capacity, so their tumor doubles faster. But also, when you look at the tumor B to T ratio, this is the ratio of the tumor cells to the effector cells, seems to be higher in the non-responder. So essentially think of it, if you have the effector cells as the cells that are going and killing the tumor cells, if that ratio is below a threshold, you may not get the response that you want. So this was interesting because the model was not trained on this. The model inferred this information. So we went back to our biomarker colleagues and said, do you have biopsies from patients and can we categorize them based on the response from complete response to partial response, stable disease, and progressive disease? So we did that and we saw exactly the same trend in our biomarker as well, that our complete response, essentially the patients that respond better, they had higher levels of T cells in their tumor microenvironment compared to progressive disease. This is something that you can essentially design or sort of like use as one of the design criteria in your V2 programs. This paper was published. This is one of the first digital twin papers that has been published in the field, and it received the ISOP 2024 Best Paper Award. So now, where are we going from here? We've used the digital twins to answer a lot of different questions for most on clinical development. Obviously, characterizing the exposure response was the first step, but now the other things that we've done is we had data from our relapsed refractory NHL patients. We wanted to look at what the CRS profile would look in the front line. So given the differences between frontline and relapsed refractory, we predicted that CRS. We wanted to change the route of administration from IV to sub-Q, and we used this in order to project the CRS risk for sub-Q and communicated with the FDA, and FDA accepted that argument. We wanted to also look at the prediction of CRS risk in sub-Q flat versus step-up dosing. We had some internal questions of what would happen if we combine mosunetuzumab with checkpoint inhibitors. So the model was used to answer those questions. Now, we've tested mosun first in relapsed refractory NHL, but we wanted to test it in other oncology and non-oncology indications such as CLL and lupus, and the model has been utilized in order to answer questions in those extensions as well. And finally, we wanted to use the models to support development of new TCBs, which is the second case study that I'm going to talk about. This one would be brief. So cevostamab is another TCB that we have in the pipeline. It's targeting FcRH5 in multiple myeloma. So we've changed the model to incorporate all the differences that we have between NHL and multiple myeloma, and essentially the FcRH5 and CD20. When we communicated the results to the FDA, they had a question for us. They were saying, okay, so you have tested single-step dosing, double-step dosing, and triple-step dosing, and you have shown simulations that triple-step dosing reduces CRS, in this case IL-6 as a surrogate for CRS, but we are not sure this is the best design. So can you use your model and digital twins in order to run a lot of different in silico clinical trials and tell us that you are exactly in the right place? So they wanted essentially to see heat maps. So we went ahead and you can see there are many different cases that you can simulate. We communicated with the FDA many different heat maps, but I'm picking here two things. So the 4 doses that you have, day 1, day 2, day 8, and day 9, you can change the timing for these doses and you can change the value for these doses. So for example, in this case, I'm fixing day 1 and day 9. I'm fixing the dose 0.3 and 160, and I'm just picking the second dose and the third dose and say, when I'm changing these doses, what would be the CRS profile and where am I with the dose that I have actually tested in patients? And that's the X here. So the first heat map essentially shows just among all the choices that you have, this dose is in a blue region and reduces the CRS compared to other scenarios. We are looking at another case if the dose was given at day 3. Again, it's not in the lowest dose, but it's in a pretty low scenario. So what we communicated was across many different scenarios that you have, this dose actually is going to give you the lowest total CRS. And FDA accepted that argument. Okay, so with that, what is next step? Obviously, we are estimating the patient trajectory and the treatment effect at the same time. We don't know how these patients would respond if there was no treatment. There are efforts now with using single-cell RNA omics data to essentially project the trajectory of the patient and then estimate the treatment response. So that's one of the exciting things that's happening. Personalized medicine, obviously, we are not there yet, but with some of the smaller, sort of like more understandable QS models, we can use them to to use it for individualized patient level response and sort of like personalized medicine. We need better uncertainty quantification methods in digital twin calibration, and obviously AI-based and agent-based digital twin generation. So with that, I want to acknowledge everyone who has contributed to this work, as well as the mosunetuzumab team, as well as the CBOS team, and all of our investigators and patients. Thank you for your attention.
Awesome. Thanks, Iraj. That was a great presentation. I guess to kick it off, I do have a question. I think you talked about kind of simulating or estimating some of the kind of observations you'll see in phase one using kind of let's say a bispecific that is kind of competing with your given context along with some preclinical data. I was curious about, especially when you're looking at kind of a different drug with the same mechanism of action, maybe you can kind of like elaborate on what are some of the considerations you take into account and how in kind of like transferring that data or learning from the other drug to kind of this model.
Yeah, no, great question. And in fact, it goes back to the structure that I'm showing here. When we were calibrating this model, we didn't have any data from phase one for our molecule because we were just at the beginning. It was first in human dose. We only had like one patient or something. But then Amgen had already published data for blinatumomab, which is the CD19 CD3 TCB. It's a different format. It's a BITE format. They have a different PK. They have different PD response. And obviously, they were going after different indications, ALL. So what we essentially incorporated, the reason that this is a good way of looking at it is QS models, they have a lot of knobs, and you can play with those knobs, and those knobs are meaningful. So we essentially went and took the approach of incorporating the PK of blinatumomab instead of the PK of our molecule. We incorporated the efficacy and the potency of their molecule within their published literature instead of our molecule. And then we also changed the disease from NHL to ALL. So given all of that, we essentially converted our model to be a blinatumomab model for ALL. So we switched off everything that we had for Mosun, and we switched on everything that we had for Blin, and we calibrated that model. Once we had done that, and we learned from their CRS profile, all we needed to do is to change the indication and change the PK back to mosunetuzumab and predict the CRS level. Is it perfect? It's not perfect, but when you don't have any data, that is essentially guiding you of where to go and where are the places that you need to be paying attention. And that was all we needed in order to start our phase one clinical trial.
Cool. Yeah. Yeah, it seems like a non-trivial task to do that, just from a mathematical perspective. So it's always
it is a non-trivial task. It's there's a little bit of art to it as well. A lot of science and a little bit of art.
Yeah, yeah. And then I think you were showing some of the plots where you were interrogating, and I guess just a reminder, please kind of like send your questions in through the Q&A or the chat if you have any. Otherwise, I could probably ask questions forever. But I think, yeah, you were kind of showing plots where you were interrogating the model around characterizing the differences, yeah, this between responders and non-responders. What I guess could you detail a little bit at a high level, like what is the actual kind of like mathematical operation you're doing with the model to kind of generate some of these plots?
Yeah, absolutely. So if we go back to the beginning of the talk, when we are creating digital twins per patient, we're not going to have one digital twin per patient. The reason for that is because there are several plausible parameter sets behind the scene that can create exactly the same response for the experimental condition you've tested, right? And that's kind of like the beauty of these models, because they can tell you like, based on what you have provided as the data points, these are plausible. We don't know which one of them is true, but if you, for example, put this patient in a different dose level, they're going to have the prediction is they're going to have a range of response. We cannot predict exactly what would happen to them. So now, given that, for any given patient, you're going to have many different parameter sets that could work, right? Obviously, it has been constrained, but those are still possibilities that they could work. So when we look at those and say, okay, now let's look at the total population and put like all of them together, 2,000 virtual patients or 2,000 digital twins. Now let's go back to the actual patient and say which one of them responded to drug and which one of them didn't respond to drug. So now I have a whole bunch of parameter sets that I can say this parameter set map to a response and this parameter set map to a non-response. That's how we are creating these profiles. So we can say among all the digital twins that we've had, what was the ratio of the B to T, the target cell or the tumor cell to the effector cell, and now map them against the responder versus non-responder. So here, the responder is the black, which suggests that the tumor B to T ratio was lower. So typically, these patients, they had a lower level of tumor to target. Tumor to effector cells. Whereas non-responders, they had more tumors and less of effector cells. So that was something that we did not teach the model. The model just learned it from the data that this needs to happen in order for these digital twins to be responders and for these digital twins to be non-responders, right? So I would say this is insight. All of these insights need to be validated and need to be communicated with the biomarker scientists and the researchers in order to see there is validity to it or not. Some of it could be just artifact of the model, and some of it could be real insights that then you can build on and then essentially design your next experiments.
Awesome. But yeah, I think it's always a nice kind of like added piece of kind of validation evidence when there's kind of an emergent property of the model that's not directly part of the fitting or objective function.
Exactly.
Confirmatory.
Exactly.
That's pretty exciting. Okay, I think we will. Oh, yeah, Isen.
Yeah, good morning, everyone. Maybe I have a little question for you, Iraj, because I'm used with data generation and synthetic data generation. And when we do such thing, we always fear the fact that one typical bias in the way we've selected the parameters to generate this population can be enhanced by the fact that we are building thousands of patients out of a little part of patients that represent this population. How do you deal with those problems? Because I assume it could be also something important in digital twins. So yeah.
No, great question. And that's also some of the things that we are always worried about. And the fact that given the limited data that you have, you may be overfitting your data to these specific actual patients and not the total population, right? So there's two things to that. One is what can you do in terms of validation and what is the actual limitation of the system, right? When a clinical team makes a decision, they are also making a decision based on these 35 patients worth of data. So essentially, we are not adding more uncertainty to what they are doing. We're saying you don't have a full 5,000 essentially patients from NHL. You only are making a decision based on 35 patients. So we're going to just enhance that decision. So that's one. We are trying to not add more to that. The second thing is like any sort of validation that we can do. And in this case, for digital twins, the validation that we came up with was kind of an orthogonal validation. We only have the digital twins being built based on one patient, right? So 50 of the digital twins are based on the first patient, the second 50 based on the second patient, and so on and so forth. But when we look at the exposure response, looking at like the figure here in 2C, we have whatever exposure that the patients have received versus the change in the tumor size. This type of a response for any given patient, they are creating just a tiny picture of this map, right? Therefore, when we are creating this map and then we are overlaying against that data, if there is a bias, you would see it in this map. Like, for example, you would see that the simulations are not going through the median of the data, or the range of like 25 to 75 would not go through that. So this would be the validation that tells you that you are not overfitting to the data and you're not increasing the uncertainty that was already in the population. But that's a great question. It took us quite a bit of time to get to this point in order to both convince ourselves as well as the clinicians that this is a good way of looking at the data and the digital twins are not an overfit.
Cool. All right. I think it's a good time to transition to your talk, Isen. And then
of course.
I think we'll also Iraj will be there for the following Q&A as well if we have.
Absolutely.
Okay, thank you everyone. Thank you, Ruben, for the introduction. So I would like today to show with you some works that we did in predicting treatment response in lymphoma using deep learning and multiple modalities. So as Ruben said before, this methodology is basically based on data, and we are using data to create some rules and be able to make prediction with that. So the first thing that I want to talk about here is how in the real life we take decision for patient. I've been part of lots of board to make a decision for the treatment of lymphoma, and these boards are multidisciplinary. It's usually called multidisciplinary tumor board, and it will drive the clinical decision. And this clinical decision for the patient to treat with one medication or another is mainly built on interpretation of images such as PET scans, biopsy. It's also interpretation of numbers, threshold from a biomarker, for example, and also interpretation of clinical record of the patient. So to help clinician making decision in this context, we usually use clinical staging and prognosis scores. So you maybe know the EP score, for example, that helps the clinician to drive this decision. They rely on basic point-based systems. So we extract information from pictures, such as TMTV or other values that are really important for the radiologist, for example. And we combine them using a point-based system to have a scoring. And these different methodology only rely on simple mathematic way of building some sums, for example, of points that best reflects what we see in this image. But what if we could have a scoring system that has an holistic overview of the patient data and that make a prediction using all available information without compromise? And this is where we started the CAR-T AI project at Dijon University Hospital, where we want to achieve this approach by using AI-based models. In fact, we have today some deep learning foundation models that can best describe medical images with really, really high details. And if we make a comparison, if we want a clinician to make the same description, it will take a lot of time and it will not be feasible in practice. So these descriptions of PET scans, biopsies, can be then combined with the clinical record and use machine learning models and like figure out some complex interaction between everything, and then we can deliver a score that best fits the patient survival and support the MTB decisions. So let's make a little focus on histology, because this is one of the part of the project where we have some initial results. And here you can see that in this example, in fact, we are using those foundation model. A foundation model is a model that was trained in an unsupervised way with thousands and thousands of slides to create, in fact, a cartography of a tumor microarray here, the one that you see on the top. And all of that in a DLBCL cohort. The work was done on a public data set prior to the investigation that we are planning at the CHU as a proof of concept. And then using this numerical description of the TMA, we were able to deploy some machine learning algorithm to predict the survival of the patient. And you can see here that with a lot of immunohistochemistry markers and also HE markers, we were able to achieve some degree of prediction for the survival of these patients. So this is clearly one of the milestones that will enable us afterward to build the multimodal scoring that we want to implement. But one thing that we should keep in mind, because a lot of time I see a lot of projects that want to make prediction using deep learning AI model. Yeah, of course, it's something that is really interesting and translational. We can discover new biomarkers, we can identify new correlation between biomarkers, and we can predict in our project, for example, the goal would be to predict the response to CAR T cells, because it's a treatment that is not cheap and has a lot of side effects. And if we can figure out if a patient could benefit from this medication or not, it will be a great thing for the healthcare. But we also have two other main interesting things that we can do with AI. One is automation. AI is really effective when we want to automate a task that human knows how to do, but it is a task that is difficult to reproduce, or a task that need to be done in a certain amount of time that the clinician don't have. And this is one of the something that is really interesting about AI. And the second one is less known, but deep learning model can help us identify new patient phenotypes and understanding the physiopathological characteristic of a disease. And it will not have a direct effect for the patient, but it can help for the future to discover new things about a pathology. And you would ask, yeah, but what about generative AI? Because I talked a lot about classifiers and methodology to make regression, but we also have some projects that are that takes profits from generative AI. And this project is a great example. It was a project that we've presented at the SFH and EIBM Congress. And in this example, we work on the Hodgkin lymphoma patient using generative AI model. And we had the HES slides, so the slides that is commonly used to study lymph node biopsies. And along with these HES slides, we had CD13 immunohistochemistry. And the idea is that it is possible that the expression of CD30, which is a biomarker in this disease and really important for the diagnosis and also other aspect of the care, would be directly linked to the aspects of the HES, the morphological aspect of the HES. And we used a specific generative model called a GAN, Generative Adversarial Neural Network, to understand the link between those two images. And what was really interesting is that we were able to reproduce a synthetic CD30 immunostaining with the HES as input of the model. So this could be really interesting for quick testing of a panel of immunostrain in live, and that with the integration of the model into a microscope equipped with a camera, so that the anapath can directly have the result that would take several hours otherwise. And finally, I would like to thank everyone that has contributed to this work. Thank you.
Thanks, Isen, for the great talk. Yeah, starting it off again. I'm curious about when you're doing this kind of, in your last example, you're doing this kind of like modality transfer from one kind of pathology imaging type to another. I'm curious about whether you've kind of put samples of that output in front of pathologists and gotten any feedback about kind of like, okay, are there any visual artifacts that seem weird or any kind of like qualitative?
Yeah. Of course, this is a great problem. And the problem is not only about just making the pathologists try to identify whether or not there are some artifacts. The real problem here also is that we don't have metrics that are reliable to assess if the generated images are correct or not. We have some metrics that helps to do that, but they are metrics that are helpful for general purpose. For example, when you see stable diffusion or other image generative models, these metrics are really interesting because they indicate if the model is really near the reality. But here, lots of characteristics are really specific for the field that we are talking about. For example, the aspects of the cell in the immunohistochemistry pictures are not really important. But the one thing that is really important for us is that is the biomarker, so the dark part of the picture, or in the correct or in the correct cells. And for now, we don't have general metrics that enables us to do that. But we can build some using segmentation algorithm and then using some calculation afterward. We can have some house-made metrics that best fit our needs. But yeah, of course, to be sure that the immunostaining, the virtual immunostaining is safe, we need to show those immunostaining to a specialist and assess if the quality is good or not. So, we don't have much more metrics that are able to do that automatically, such as we have, for example, for segmentation in CT scan, TEP scan. We have good metrics that with the prior annotation, we can have a good overview of if the model is performing well or not. For this type of application using generative AI, it's much more difficult.
Cool. Yeah, and I guess another question I had, because I'm less familiar with kind of digital pathology myself, but I think when we're talking about kind of bulk RNA-seq and single cell RNA-seq, batch effects are a big problem. And I think, with imaging as a modality, it might be slightly less of a problem. I don't know if any of the staining methods have similar kind of like batch or kind of like, okay, these different labs process things differently. And then on kind of like when you're doing prognostication, let's say, I imagine different kind of providers have different kind of like treatment plans and things like that, which might affect kind of your endpoints and your signal that you're fitting to. So I'm curious, kind of, are there any things that you have come across where like, okay, when you start to generalize across facilities or sites or anything like that, there are some real challenges there?
It's always the case. In fact, when you build an AI driven application, when you want to deploy the software, it's always you always have lots of work to do in the institution itself to deploy the model. And usually we worked, for example, for recently in an AI model that could predict whether a cell is a ring sideroblast or not. This exam is settled for patients with myelodysplastic syndrome, so a type of blood cancer. And the fact is, we were able, using an algorithm, to make a classification of those cells, and the classification is really difficult even for the clinician. So that's why we wanted to do that. And the thing is, to deploy this specific algorithm, we need to talk with the colorant manufacturer, for example, to have exactly the same colorant for the institution or the centers that will acquire the AI solution. This is something that can be a little bit difficult, but we have some techniques, we have data augmentation that can leverage those problems. We also have some out of distribution techniques. We can also use GAN software to correct a little bit the distribution in the clients that want to deploy the software. But it's always a challenge, this part. Yeah.
Cool. Iraj has a question. Go ahead.
Yes, and thanking you so much for the nice presentation. I had a question about the Kaplan-Meier curves that you had in the next slide.
Yeah.
Can you walk us through how you first of all, is this retrospective analysis? And then is this something that can be done prospectively, like incorporating it as sort of like a marker or digital biomarker, in fact, in a clinical trial? So I wanted to hear your thoughts on that.
Yeah, in fact, we have a way of taking the problem. First, what I usually do is that I work a lot on digital libraries, data set that are freely available to test some hypotheses. This work specifically is the beginning of the work that we did on a public data set prior to the clinical trial. And then the idea was to try and see if we can reproduce the experiment using those different public data sets, so that we have a clear insight of whether or not the methodology, but also what we want to test in our clinical settings can be reproducible and has also been shown in the literature. And then afterward, yeah, we will do a retrospective clinical trial and a prospective clinical trial. The third step afterwards will be to have a multicentric clinical trial to test the model that we were able to conceive during the initial trial with the 96 patients injected with CAR T-cell and with the model that we were able to produce. And then I have a test set from a center that is outside the center that was used initially to build the model. This is, I think, there's something that is really interesting, is that developing an AI model is, because I'm a pharmacist, and it's pretty much we can make a parallel with the development, for example, of a medication. And this is quite interesting because when you have a data-driven AI model, it will likely behave like a bioengineering project. We don't really know exactly how the model will behave. So we rely on statistics, we rely on generalization analysis to avoid the risk for the patient. And then we make some benefit-risk balance with the model. So yeah, I don't know if it was...
Thank you.
But no, I completely agree. I think I was helping a CRO kind of like start introducing AI workflows to this is completely unrelated to the topics, but just introducing AI workflows to their own kind of like literature reviews and all of these things. And I made the same kind of like corollary where I was like, just treat it treat it as kind of like a clinical development plan where you test it on a couple of different workflows, you see what happens, you go in and do like full inspections of what happened, and then you start to like slowly scale up what goes in and out of the black box, and you kind of like gain confidence in what it's doing as you scale and observe. But yeah.
And
Okay.
then you also have like surveillance of the model afterward, like pharmacovigilance. In fact,
Right.
we can also make the parallel with that. And we've developed also some AI model. Now we have the ability to identify if a model is failing. That's really important because lots of clinicians told me, yeah, we don't want the model that is the most accurate, the model that is able to make the exact precision, like survival prediction for the patient, because this won't really change the way that we are going to treat the patient. But the most important is to have a model that knows when it is failing and like launch an alert to tell, yeah, actually the model is failing because the data that you're giving the model is not like similar to the training data. And this can be done with lots of different implementation. For example, we've worked a lot with Bayesian methods where you had some Monte Carlo dropout to the model so that the model can like detect some out of distribution or aleatoric epistemic uncertainty, and then give all of this information to the practitioner so that he can take a decision and say, yeah, for this analysis, I can validate just by a simple check, and the other one I need to do it.
Oh, I think we lost your audio somehow.
Yeah,
There you go.
it's okay. Yeah, thank you.
Cool. Yeah, I agree. It's actually like a surprisingly difficult and multidisciplinary kind of problem to kind of monitor and estimate how your model's doing once you release it. But okay, I think with that, we can kind of wrap up. Thanks again, Iraj and Isen, for the awesome talks. They were really awesome and thorough kind of deep dives into different computational methods. So it was quite interesting. Um, Thanks again for the participants here. I think you should be receiving a survey for feedback, so please fill that out to kind of keep improving the talks. The next episode of Red Talks will be season 6, episode 5, about bispecifics and CAR T cells, a practical guide to navigate the new landscape, on the 9th of September at the same hour. And yeah, thanks again, everybody.